Hi, I would like to thank the organizers for the opportunity to present our proceedings paper, Fast Approximate Shortest Hyperpaths for Inferring Pathways in Cell Signaling Hypergraphs. This work was done by Spencer Krieger and John K. Sisioglu. I'm Spencer Krieger, and I'm excited to share this work with you today. Cell signaling is how cells exchange information with their environment by sending and receiving small molecules at membrane-bound receptors, which is fundamental to cell biology. A cell signaling network encompasses all the chemical and physical reactions that occur in the cell. A cell signaling pathway is a subnetwork of these reactions that describes a cellular process and usually begins at one or more membrane-bound receptors and ends at transcription factors and regulators. Here I show an example of the p53 signaling pathway, which is involved in DNA repair and apoptosis. As shown on the left, when a stress signal is sent, p53 promotes DNA repair and downstream apoptosis by aiding in the transcription of several proteins. However, shown on the right, when there is no stress signal, p53 is inactivated and degraded when it's bound to MDM2. Perturbations in this p53 pathway causes cancer. Notice the series of reactions when p53 is deactivated by MDM2, which starts at p53 in the cell and ends with p53 and MDM2 being transported from the cell as a target reaction. Inferring the most likely series of reactions, or pathway, from a given set of receptors or sources to a given transcription factor or target is a key task. Cell signaling pathways are made up of a series of cascading reactions of this general form, with reactants on the left, here shown as A through B, and products on the right, here shown as C through D. Biochemically, the reaction will only proceed when all reactants on the left of the arrow are present, and when the reaction occurs, all of the products on the right are produced. Determining signaling pathways biologically is laborious, so we infer them computationally. Therefore, we need an appropriate graph representation of signaling networks. Previously in the literature, ordinary graphs have been used to model cell signaling networks. The previous reaction I showed is modeled like this, with directed edges connecting each reactant to each product. The issue is that edges can only capture pairwise interactions, not multi-way reactions, which is what we want. In this representation, you can reach product C without ever reaching or producing reactant B. So there is no guarantee that all the reactants are present, or even that all the products are produced. This shows that ordinary graphs are insufficient to represent signaling networks and pathways. We use directed hypergraphs, which are a generalization of ordinary graphs, to correctly model these cell signaling pathways. A directed hypergraph consists of vertices and directed hyperedges, where a hyperedge generalizes an ordinary graph edge and is a pair of vertex subsets x and y, where now the edge goes from a set of vertices, its tail, to a set of vertices, its head, as shown here in the picture. To traverse edge E, all of the vertices in the tail, here A through B, must already be reached, and upon traversing the edge, all vertices in the head, here C through D, are reached, like in the reaction I showed on the previous slide. In a cell signaling hypergraph, vertices represent molecules, including proteins, and hyperedges represent reactions. A specific signaling pathway is a series of hyperedges from source vertices to target vertices, which is formalized as a source-sink hyperpath. This hyperpath goes from S to T. Notice on the left that there can be multiple out edges from S. In the middle, hyperedge E highlights a general hyperedge, where all of its tail vertices must be reached by prior hyperedges in the hyperpath, like hyperedge F, for E to be traversed. And on the right, T is one of the vertices in the head set of the final hyperedge. More formally, we define an ST superpath, different from a hyperpath, for source S and sink T as an edge subset F where the hyperedges in F can be ordered, such that the tail set of the first hyperedge contains only the source vertex, which captures that the superpath starts from S. For each hyperedge E sub I, its tail is contained in the union of the source and the headsets of the preceding hyperedges. And finally, that the sink is in the head of the final hyperedge, which captures that the superpath ends at T. Informally, sink T is reachable from source S if there is an ST superpath. Finally, we define an ST hyperpath as a minimal ST superpath. Minimality captures that there are no useless hyperedges in the hyperpath. In other words, in removing any hyperedge, T is no longer reachable from S. The shortest hyperpaths problem is, given a hypergraph, a positive edge weight function, a source vertex S, and a sink vertex T, find an ST hyperpath of minimum total weight. The shortest hyperpath is the most parsimonious series of reactions that produces the targets from the sources. In many cell signaling databases, hyperedges have unit weight, which corresponds to finding the fewest reactions to produce the target from the source. Shortest hyperpaths is NP-complete, even for acyclic hypergraphs where all hyperedges have singleton headsets. The NP-completeness proof is from set cover and is approximation preserving, so shortest hyperpaths is similarly inapproximable to within a ratio of 1 minus little o of 1, ln of n. Our definition of shortest hyperpaths, which contains only a single source and a single sink, is also general, as the multiple source, multiple sink problem can be reduced to the single source, single sink problem in the following way. Given a set of source vertices, which we label with S1 through Sk, we can create a supersource which is connected to the sources by one hyperedge. Then, for the multiple sink vertices, T1 through Tl, we can do the same and connect them to a supersink by one hyperedge, which requires all sink vertices to be reached. However, if you only want to require that at least one sink vertex is reached, you can connect the sinks T to the hypersink individually with ordinary graph edges. The current state of the art for shortest hyperpaths in cell signaling networks finds only acyclic hyperpaths by solving a mixed integer linear program, or MILP. Recall that even the acyclic problem is NP-complete. And because the MILP solves this NP-complete problem, it does not have any efficiency guarantees, but is practical. Integer variables here capture the hyperedges in the hyperpath, and real valued variables enforce an ordering on the vertices in the acyclic hypergraph. This formulation has no natural extension to handle cycles. Feedback cycles, however, are ubiquitous in signaling networks, and different from ordinary graphs, shortest hyperpaths can contain cycles, as shown here, where this ST hyperpath contains a cycle, shown in red by the single hyperedge which has a previous vertex from the hyperpath in its head. In practice, where all hyperpaths from S to T are cyclic, the MILP finds no solution, even though ST hyperpaths exist. In this work, we give the first efficient heuristic for general shortest hyperpaths that can contain cycles. Our heuristic is efficient. For a hypergraph of size L, which is the total cardinality of all head and tail sets, with M hyperedges in a doubly reachable subgraph, which I explain on the next slide, of size K, the heuristic runs in big O L plus M squared times K time. In practice, the doubly reachable set is much smaller than the full hypergraph, so the superlinear portion of the computation is over a small subgraph. We also give a simple and practical hyperpath enumeration algorithm, which allows us to tractably measure how close our heuristic is to optimal. Experimentally, our heuristic matches the state of the art on over 99% of all acyclic instances from 2 standard cell signaling databases. And when every hyperpath is cyclic, the MILP finds no solution. So our heuristic surpasses the state of the art on cyclic instances where no acyclic hyperpath exists. We verified that our heuristic is optimal in all such cyclic instances by our hyperpath enumeration algorithm. Now for a few definitions. Here is a formal definition of the size of a hypergraph G, which is the total cardinality of all tail and head sets of the hyperedges in the hypergraph. We define an outedge f of hyperedge E as any hyperedge whose tail intersects the head of hyperedge E, as shown here. We define a hyperpath from source vertex S to hyperedge E as a hyperpath where each vertex in E's tail is reached by the head of at least one hyperedge in the SE hyperpath. We call hyperedge E forward reachable if an SE hyperpath exists, and we call a hyperedge backward traceable if its head contains T, or if the hyperedge's head intersects the tail of a hyperedge that is backward traceable, and so on. We call the hyperedge doubly reachable if it is both forward reachable and backward traceable, as E is here. The doubly reachable subgraph is the intersection of the forward reachable hyperedges and the backward traceable hyperedges. To preview our shortest hyperpath heuristic, I will start by introducing Dijkstra's shortest path algorithm for ordinary graphs, which will then be contrasted to our heuristic, which proceeds in a similar manner, but with key differences. Dijkstra's algorithm finds a shortest ST path in ordinary graphs. It maintains a heap of vertices, prioritized by their estimated distance from the source as their key, and each vertex holds on to its best inedge. The heap is initialized with the source vertex S, and until it is empty, we repeatedly extract the vertex V with the smallest key, which is the length of the shortest SV path. Then, for each outedge of V, edge VW, we update W's key with the length of the best SW path we have so far. And then we hold on to the best inedge to W. Finally, we recover the shortest ST path starting from T using the best inedges stored in step 3. Similarly, our heuristic finds an approximate shortest ST hyperpath. The heuristic also maintains a heap, but it is a heap of hyperedges instead of vertices, with an edge's tail distance as the key, which is the length of the best known hyperpath from the source to the vertices in the tail of E. We keep a list of inedges, as a hyperpath ending at a tail set consists of many final hyperedges. We initialize the heap to the hyperedges with the source in their tail, and repeatedly extract the hyperedge with smallest key, which is the smallest estimated tail distance. Then for each outedge f of E, we update f's inedge list with E. We then use a greedy heuristic to approximate the shortest hyperpath from S to the tail of f, which is also NP-complete, and update f's key if this approximate shortest SF hyperpath is shorter than previous ones. Then, we again use this greedy heuristic to recover an approximate shortest ST hyperpath. Our shortest hyperpath heuristic is composed of a few component algorithms. First, forward reachable, which finds the set of hyperedges that are forward reachable from the source vertex S and runs in time linear in the size of the output hypergraph G. Next, backward traceable, which similarly finds the set of backward traceable hyperedges from the sinc T in time linear in the size of G. Finally, recover short hyperpath, which is a greedy heuristic that finds an approximate shortest hyperpath from the source to an input hyperedge E, which will be repeatedly called in the shortest hyperpath heuristic to give approximate tail distances for hyperedge E. The greedy heuristic also makes use of estimated tail distances that have been computed previously. Next, we will go into a little more detail of this greedy heuristic. More concretely, recover short hyperpath, which greedily finds a short SE hyperpath, has two steps. First, it recovers an SE superpath P, starting from final hyperedge E and backward traversing the inedges of E back to the source, using the inedge list that have been populated by the overall heuristic. Second, it greedily trims P to a minimal SE hyperpath in the following manner. It first sorts the hyperedges in P by their estimated tail distance. Recall that the estimated tail distance for E is the length of the shortest known hyperpath from S to the tail set of E. Then, In this sorted order, it considers removing hyperedge f from the superpath if E is still reachable from the source upon removing f from the superpath. Removing the hyperedges in this order is likely to leave the shortest hyperpath, because redundant hyperedges with higher tail distance will be deleted before redundant hyperedges with lower tail distance. The time complexity of recover short hyperpath is big O m sub h times the size of h, where m sub h is the number of hyperedges in the doubly reachable subgraph h. Now going back up to the overall shortest ST hyperpath heuristic. It first computes the doubly reachable subgraph of G, taking the intersection of forward reachable and backward traceable. Note that it is not worth looking at hyperedges that are not forward reachable and not backward traceable, since they can never be in any ST hyperpath. The heuristic maintains a heap of hyperedges, which are prioritized by their estimated tail distance. Then, until the heap is empty, we extract hyperedge E and call recover short hyperpath to get an updated estimated tail distance for E. Then, for all outedges f of E, we call recover short hyperpath to get an improved estimated tail distance for f. Now that E has been given its final estimated tail distance, using f's updated inedge list in recover short hyperpath may improve its estimated tail distance. Finally, once all hyperedges have been removed from the heap, we call recover short hyperpath on the inedges to the sink to recover the best ST hyperpath. Notice here all of the calls to recover short hyperpath in our heuristic. As shown in the previous slide, recover short hyperpath is called many times, so it dominates the running time of the overall heuristic, being called at most m sub h times, where m is the number of hyperedges in the doubly reachable subgraph h. Overall, the time complexity of our heuristic is big O, size of G, which is from computing the doubly reachable subgraph, plus m squared h times the size of h, which is from the repeated calls to recover short hyperpath. In practice, our heuristic runs in under a minute for the largest instances of our datasets, which have around 10,000 hyperedges. To evaluate the quality of our heuristic, we need to find the length of the shortest hyperpath, which we do by enumerating all hyperpaths using our new enumeration algorithm. To give an overview, it maintains a queue of subproblems, which is based on outsets. Solving a subproblem, which requires certain hyperedges to be in a hypergraph, is an NP-complete problem, so we do not require certain hyperedges to be in the solution to a subproblem. When we solve k subproblems, the running time is big O k times m times the size of the hypergraph G. In the worst case, there are subproblems exponential in the number of hyperedges in the graph. The algorithm takes many hours to run, but it enables tractable enumeration of all hyperpaths, even for large instances. We now move to experimental results. To determine the quality of our heuristic in practice, we constructed hypergraphs using human-curated signaling pathways from standard pathway databases, NCIPID and Reactome. We parsed these datasets into hypergraphs using a parser from Francesi et al., which was built on PAX tools. We map each entity, be it a protein or small molecule, to a vertex, and we map each instance of that entity to a different vertex, so the same protein in different cell compartments or with different post-translational modifications is represented by a different vertex. We then also map each reaction to a hyperedge. Any vertex with no inedges is used as a source in our shortest hyperpath instances, and any vertex with no outedge is used as a target. We added a single hyperedge from a supersource to all source vertices, and for each target instance, we have a problem instance, which corresponds to finding a shortest hyperpath from all sources to this target as the sink vertex. Note that in our hypergraphs, all hyperedges have unit weight, as reaction rates are not available for all the reactions in the databases. We summarize the size of our hypergraphs in the following table. The hypergraphs from both datasets are very sparse, with each dataset containing more vertices than hyperedges. The hypergraphs are composed of the union of many different pathways. with each hypergraph containing nearly 1/3 of their vertices as sources, or vertices with no in-edges. For each dataset, there is also a comparable amount of targets, giving us our over 2,600 problem instances on NCIPID and our over 5,000 problem instances in Reactome. I will now share results on the acyclic instances from our hypergraphs, which are instances where there is an acyclic ST hyperpath. Of the reachable instances for each dataset, most are acyclic, as seen when you compare the top two numbers of each column. On over 99% of the instances, our heuristic matches the state-of-the-art MILP, while providing a guarantee of an efficient running time, which is not provided by the MILP. In the few instances from NCIPID where the heuristic was suboptimal, it was very close to optimal. The median difference in hyperpath length, which is the number of hyperedges in the hyperpath, between the heuristic and the shortest hyperpath from the MILP was only one hyperedge. In cyclic instances, where all ST hyperpaths are cyclic, the MILP returns no solution because no acyclic ST hyperpath exists. Looking again at the reachable target instances, we see that there are many cyclic instances. In all of these cyclic target instances, our heuristic was optimal, which we proved by enumerating all hyperpaths, choosing the shortest one, and comparing its path length to the heuristic. We also show that the heuristic is discriminating between different hyperpaths, as there are 50 hyperpaths on average for NCIPID with a large range between the length of the shortest and longest hyperpaths, which difference was 9 on average for NCIPID and reached as high as a difference of 43. The number of enumerated hyperpaths is slightly smaller for Reactome because its hyperedges are even sparser than NCIPID's. We point out that for every cyclic target instance, our heuristic returned an optimal shortest ST hyperpath. I will now highlight one of these cyclic instances so you can get a feel of what these shortest hyperpaths look like. Here we show the shortest hyperpath to the phosphorylated p53 tetramer, which shows the activation of p53 in the cell. Here, the sources are along the top of the figure, which are the vertices in the hypergraph with no in-edges, so we assume that they are already present in the cell. The target is on the right, the phosphorylated p53 tetramer. Hyperedges E2 and E3 are dashed because they show transcription of a protein, and the small circle with a P inside shows phosphorylation. So here, transcription factors FOXO3 and FOXO4 facilitate the transcription of STK11 in hyperedges E1 and E2. P53 is transcribed and forms a homotetramer in hyperedges E3 and E4. This tetramer then forms a complex with ARK5 and STK11 to allow phosphorylation in hyperedge E5. Then ARK5 is phosphorylated in E6, and the P53 tetramer is phosphorylated in E7, breaking the complex and creating a cycle because STK11 is both in the head of hyperedge E7 and in the tail of hyperedge E5. In this talk, I have presented the first heuristic for the general shortest hyperpath problem that properly handles cycles instead of only allowing acyclic hyperpaths, that is guaranteed to be efficient instead of having possible exponential running time, and that matches the state-of-the-art MILP on 99% of all acyclic instances, and that surpasses the state-of-the-art on all cyclic instances, where the state-of-the-art returns no solution. And we have shown experimentally that the heuristic hyperpath was optimal. Source code for our heuristic, along with the datasets used here, is available at hyperpaths.cs.arizona.edu. There are a few avenues of further research on the shortest hyperpaths problem. From the NP completeness reduction for shortest hyperpaths, we know that any approximation algorithm must have an approximation ratio of at least log of n, where n is the number of vertices. Does there exist an approximation algorithm whose approximation ratio matches this known lower bound of log of n? Experimentally, our heuristic was only suboptimal on acyclic instances. We would like to know if there is an efficient heuristic for the acyclic version of shortest hyperpaths that improves upon our heuristic. In practice, a user running a heuristic would like to quickly know how close the hyperpath heuristic is to optimum. Is there an efficient heuristic that outputs a lower bound on shortest hyperpath length so we can quickly determine the quality of the heuristic hyperpath without needing to enumerate all hyperpaths, which can take many hours? We would like to thank T.M. Morale for introducing us to cell-signaling hypergraphs, and Anna Ritz for discussing the NCI PID and Reactome databases. We also thank the U.S. National Science Foundation for funding. I will be graduating in the spring of this upcoming academic year, and I'm interested in postdoc positions. Thank you for listening to my presentation.